MS Ramaya University of Applied Sciences.
Hi students, study of appliances used in hematological experiments. Aim of the experiment: to study the various appliances used in hematological experiments and to utilize the right appliances to perform hematological experiments. Introduction: hematology is the study of blood, blood forming organs and blood diseases. Hematological experiments involve the examination and estimation of blood and its components such as blood cells, hemoglobin, blood proteins and coagulation. These experiments assist in the diagnosis and ultimately in the treatment, prognosis and prevention of blood diseases. The purpose of this experiment is to impart knowledge about various appliances used in hematological experiments. Appliances used in hematology: hemocytometer, hemoglobinometer, Winthrop tube. Hemocytometer: it is an equipment used for counting the number of cells, RBC or WBC, in a sample of blood. The hemocytometer set consists of the diluting pipettes and Neubauer's chamber as shown in the figure. The diluting pipettes: two different glass capillary pipettes are used for counting red blood cells and white blood cells. These pipettes are also called as cell pipettes or blood pipettes. Each pipette has a long, narrow stem with graduations which widens into a bulb that contains a bead for mixing and diluting of the blood, white bead in case of WBC pipette and red bead in case of RBC pipette. The bulb narrows again into a short stem bearing a soft rubber tube and mouthpiece, red in case of RBC and white in case of WBC pipette. Just beyond the mouthpiece, the number 101 is etched on RBC and 11 on the WBC pipette. The counting chamber, improved Neubauer's chamber. The counting chamber is a single solid heavy glass slide. The floor piece is divided into two equal parts by a short transverse H-shaped trench in its middle. Identically ruled areas called counting grids consisting of squares of different sizes are etched on each floor piece. Counting grid of Neubauer's chamber. It has following dimensions. Each counting grid measures 9 mm square. It is divided into 9 large squares of each 1 mm square in dimension. Different counting areas in Neubauer's chamber. Of these 9 squares, the 4 large corner squares are lightly etched and each is divided by single line into 16 medium sized squares. These 4 squares are employed for counting leukocytes and are therefore called as WBC squares. The central densely etched large square, which is of 1 mm square in dimension, is called RBC square and is divided into 25 medium sized squares. Each of the 25 medium sized squares are bound by triple lines and is further divided into 16 smallest squares by single lines. RBC and WBC counting areas can be seen as shown in the figure. Hemoglobinometer or Shally-Adams hemometer. It is an equipment used for estimation of hemoglobin in blood. The set consists of the following: hemoglobin comparator, hemoglobin tube, hemoglobin pipette, Pasteur pipette, stirrer, 0.1 N HCl solution and cleaning brush. Components of hemoglobinometer: hemoglobin tube. The square or round tube is calibrated in grams percentage in yellow color on one side and in percentage in red color on the other side. Hemoglobin pipette. It is a glass capillary pipette with only a single calibration mark that is 0.02 ml or 20 microliter, there is no bulb in this pipette as no dilution of the blood is done. Stirrer. It is a thin glass rod with a flattened end which is used for stirring and mixing the blood and dilute acid. Pasteur pipette. It is an 8 to 10 centimeter glass tube drawn into a long thin nozzle and has a rubber teeth. Cleaning brush. It is used to clean the hemoglobin tube. Components of hemoglobinometer are as shown in the figure. Winthrop tube. It is used for the estimation of erythrocyte sedimentation rate and hematocrit. It is 11 centimeter long, heavy cylindrical glass tube with a uniform bore diameter of 2 mm. Its lower end is closed and flat. It is graduated 0 to 10 centimeter from above downwards on one side for ESR and 10 to 0 centimeter on the other side for the hematocrit. The Winthrop stand can hold 3 to 6 tubes at a time. It is provided with a spirit level to ensure that the tubes are held vertically throughout the test. Other appliances used in hematology. Cover slip. Special cover slips having an optically plane and uniform surface are preferred over ordinary cover slips. Blood lancet. It is a small medical implement used for capillary blood sampling. It is used to make punctures such as finger stick to obtain small blood specimens and generally disposable. Hi students, in this experiment we are going to study the various hematological appliances used in hematology. The aim of this experiment is to understand the different components and the parts of various appliances that are used in hematological experiments. Hematology is the study of blood forming organs and the diseases which are associated with blood. Hematological experiments deals with examination and estimation of blood and its components such as blood cells, coagulation factors, etc. These experiments helps us to understand, diagnose the blood and the diseases which are associated with it and ultimately assists us in prevention of these diseases. Now let us study the various appliances used in hematology. The various hematological appliances are RBC pipette. This is one of the diluting pipette used in hematology. This pipette is called as RBC diluting pipette, which is utilized to dilute the RBC cells in the blood. It consists of a narrow stem which is having various graduations to measure the blood. This narrow stem further widens as a bulb which consists of a red bead to mix the blood with the diluting fluid. Further, this bulb continues as a narrow stem, and to this narrow stem the soft rubber tube is attached. At the end of the rubber tube, a mouthpiece is provided to suck the blood and the diluting fluid. In case of RBC pipette, this mouthpiece is red in color, and slightly above this bulb the mark 101 will be etched in case of RBC pipette. WBC pipette. This is one more diluting pipette which is used for WBC. This is also called as WBC diluting pipette. It also consists of a narrow stem with various graduations to measure the blood. Then the narrow stem widens as a bulb which contains white bead for mixing of the blood with the diluting fluid. Further, this bulb extends as a narrow stem to which the rubber tube is connected with the mouthpiece, and this mouthpiece is white in color in case of WBC pipette, and slightly above the bulb the marking 11 is etched in case of WBC pipette. Neubauer's chamber. This is improved Neubauer's chamber or the counting chamber. The Neubauer's chamber is a single solid heavy glass slide. The floor piece is divided into two equal parts by a short H-shaped trench in its middle. Identically ruled areas called counting grids containing of squares of different sizes are etched on each floor pieces. The counting grid has following dimensions. Each counting grid measures 9 mm square. It is divided into 9 large squares of each 1 mm square in dimension. Of these 9 squares, the 4 large corner squares are lightly etched and each is divided by single into 16 medium sized squares. These 4 squares are employed for counting leukocytes and are therefore called as WBC squares. The central densely etched large square is called RBC square, which is further divided into 25 medium sized squares. Each of the 25 medium squares are bound by triple lines, which is further divided into 16 smallest squares by single lines. Hemoglobin tube. This is hemoglobin tube which is utilized in measuring hemoglobin. It consists of two different types of graduations, one in yellow color which indicates gram percentage of hemoglobin. One more is in red color which indicates percentage of hemoglobin in blood. Hemoglobin pipette. This is hemoglobin pipette which is utilized in estimation of hemoglobin content of the blood. It's a glass capillary pipette with only a single calibration mark of 20 microliter on it. There is no bulb in this pipette as compared to the cell pipettes that we studied earlier, as no dilution of the blood is done. The stem of the pipette further continues with a soft rubber tube which is attached with a mouthpiece which is white in color. Pasteur pipette. This is pasteur pipette. It is an 8 to 10 inch glass tube which is drawn into a long thin nozzle and it has a rubber bulb. Stirrer. This is stirrer. It is a thin glass rod with a flattened end which is used for stirring and mixing the blood with the diluted acid. Winthrop tube. This is Winthrop tube which is utilized in the estimation of erythrocyte sedimentation rate and hematocrit. It is a glass tube of 300 mm in length and consists of graduation from 0 to 200 mm. And hemoglobin comparator. This is hemoglobin comparator. It is a rectangular box with a slot in the middle which accommodates the calibrated hemoglobin tube. Non-fading standardized golden brown glass rods are fitted on each side of the slot as standard for matching the color while performing the experiment. An opaque white plastic glass is fitted behind the slot to provide uniform illumination during direct visual color matching. With this, we complete the study of various hematological appliances used in hematology.
At MSR UAS, we spark your imagination unlike any other.